Good afternoon, everyone. Thank you for taking the time to come over for this session. I'm sure you guys have a lot of questions. It's a very burning clinical thing, genetics in pulmonary fibrosis and what is important. So I'm glad we have two excellent speakers who are going to talk about the science and the clinical aspects of it, and obviously the genetics and the whole spectrum. And as you can imagine, the focus is predominantly to make it relevant to the patients, and that's what we're going to aim for. So we're going to let them finish the talk, and then we're going to please note down your questions, and we're going to have a whole Q&A after that. And the speakers have promised me they're not going to go beyond 45 minutes, and probably it's going to be shorter, so we can have a lot of questions we can answer at the end of it. And I know I'm the only one who's between you and cocktail hour, never a good thing. But somehow I always get the short straw, I guess. My genetics, it's very, very evident when you look at me. It's not exactly European style. So, but anyway, jokes aside, we have Dr. Kropski, very phenomenal investigator, actually. I had a chance to look at his CV when I was called in to moderate this session. Excellent work, a lot of information. I'm sure he's going to update us with all the scientific work which he has done about genetics. He's a great writer. Some of the editorials I've seen him writing in different journals, and I'm sure I'm actually myself looking forward to his talk. So why don't we give a warm welcome to Dr. Kropski.
Can everyone hear me okay? Okay. Great. Well, thank you all for being here, and thank you for the chance to discuss a topic that I've spent a lot of time thinking about, and a topic that is very near and dear to my heart. I have to say, I did not know anything about pulmonary fibrosis until I was a first-year medical student, and my best friend in medical school's father was then diagnosed with pulmonary fibrosis, and then so was his uncle and cousin brother. So this has been sort of a personal as well as a professional interest now for a long time. Over the course of hopefully about the next 25 or 30 minutes, I'd like to talk to you about genetics broadly and sort of introduce how we've been approaching trying to understand genes in families with pulmonary fibrosis and how we should think about it, both in terms of caring for patients who have pulmonary fibrosis and what the implications are for their families. And then I'm going to turn things over to Janet, and she's going to, I think, really take a deeper dive into understanding how genetic testing and genetic counseling works and how that works in individual families. These are my disclosures. None of them are really relevant to this particular talk. So to start with, I'd like to give just an overview of familial interstitial lung disease. I'd like to give a little bit of an introduction to how we inherit risk, then talk a little bit about genes that have been linked to pulmonary fibrosis in families. Then I'd like to give you a little bit of an update as to how all of this work has helped us understand how these diseases evolve through time and what we've learned about the basic biology of pulmonary fibrosis. And then last, what does this all mean for us? How should we use this information? So to start with, pulmonary fibrosis is a genetic disease. And the reason that we know that is that if you have a family history of pulmonary fibrosis, that is the single strongest risk factor for developing this disease. So someone who does have a family history is at least 6 times more likely than someone that doesn't to develop pulmonary fibrosis. And that effect it's stronger than smoking. It's stronger than any of the other occupational exposures that people may be exposed to. So heritability really matters in this disease. And so people have tried to understand how that works really in 3 different contexts. First, in families where there are multiple people who are affected with interstitial lung disease, and we call that syndrome familial interstitial pneumonia or familial pulmonary fibrosis. Second, there have also been studies trying to understand how genes confer risk for this disease in people who don't have a known family history of interstitial lung disease. And third, there are also some syndromes that affect multiple different organ systems that have interstitial lung disease as a component of them. So these are things like Hermansky-Pudlak syndrome, dyskeratosis congenita, and a couple of other less frequently observed syndromes with names like COPA and STING, which are the genes that are mutated in them. These tend to present more in childhood, so I'm not really going to focus on that here today, but I'd be happy to talk about those more if anyone has specific questions. So a point that I think is really important, and it's an important point for the people who take care of pulmonary fibrosis patients, but it's also important for people and families, is to recognize that not everyone within a family is going to have the same type of pulmonary fibrosis. And so on let's see if I can do this without, I'm not sure if is there a laser pointer with this? No, okay. On the left-hand part of the screen there are just two different CAT scans from two different individuals in the same family. And while it's clear that they both have fibrotic interstitial lung disease, one has much more peripheral, this is tricky, peripheral reticulation and honeycombing, and one has quite a bit more ground glass. And and we've recognized this now for quite a long time. So really the first study that showed this was one that Dr. Mark Steele published now almost 15 years ago from a registry at Duke and at Vanderbilt that showed that about 50% of families have different diagnoses, so different patterns of disease in them. And this holds true even in families where you do have a known culprit gene that's responsible. So one thought that we had through time was that different genes are going to lead to different specific pathologic patterns, so different sort of injury patterns on slides or different imaging patterns, and that hasn't really turned out to be true. And so the reason that I think that this is important is that it suggests that genetic susceptibility is kind of an underlying mechanism, and it may not matter quite so much what other genetic or injurious factors are. That may not be what really drives someone's chances of getting this disease. Okay, so what does confer genetic risk? As a very, very brief introduction to genetics, for those who don't think about this all the time, we as humans have 23 pairs of chromosomes. That means you have 2 copies of each gene. And we have about 20,000 different genes that encode for a protein. And so it turns out that we all have variations, changes in that DNA sequence that differs from what's called a reference. So reference genomes are things that are built and assembled by analyzing large groups of people and essentially defining what's the most common sequence at a given site. So everyone has genetic variants. Another thing that is important to recognize is that almost all of our DNA doesn't encode for a protein. So proteins are the workhorses of our cells. They are structure, they're enzymes, they do much of the business, but the vast majority of the DNA in our genome doesn't encode for proteins. And so what we think that does is that it's involved in regulating when and how the protein encoding parts of the genome act. But there's really a tremendous amount of knowledge that we need to learn about what all of this, what we call non-coding DNA does. And the reason that that's important is that right now we don't have very good ways of knowing whether sequence changes in 98.5% of our DNA matter or not. And they might, but this is an entire largely unexplored area that we're just getting into right now. Okay, again, going back to, this didn't project particularly well, but just thinking how does a mutation in a gene impact what a cell can do. So our DNA, the process that we get from DNA to proteins is that our DNA, again, we have a couple copies of it, it unfolds and it undergoes a process called transcription, where the DNA is basically copied into a different molecule called RNA, and that RNA is then processed into this single continuous message. Now, it turns out that when this happens, both parts of our genome that encode for proteins, as well as parts that are some of those regulatory regions, they all come up off in sort of one big strip, and so this has to get processed properly in order to get a functional protein. And so then the proteins, or the messenger RNA intersects with these things called ribosomes, and then they build the proteins that are the functional units of our body. So the ways that a genetic variant can lead to a dysfunctional protein are basically all of the ways that something in that process can go wrong. So for example, you can have what's called a premature termination codon. So that is like a stop sign in your DNA. So you have code in your DNA that tells those enzymes that are making protein to stop acting. So you can basically have a stop sign too soon. That means you don't make any functional protein. You can also have sequence changes that affect how you can stick those pieces together while they're getting processed. And so if you don't stick those pieces together properly, then you end up with something missing or something duplicated and you have a protein that doesn't work right. You can also have things called insertions or deletions. So these are places where you have like a chunk of the DNA that either gets inadvertently copied multiple times, or someplace that some chunk of it doesn't get copied properly, it's missing, and so the pieces around it get stuck together. And so that can also then lead to a change in the composition of a protein, and that frequently leads to changes in its function. And then the last type of genetic variants that I want to mention are things called single nucleotide variants. So that means a single base, one letter in your DNA code is changed. And these can be innocuous, or they can be they can have significant functions. So sometimes they might change the DNA code, but it doesn't change what the end product of the protein is. So we call those synonymous. So those almost always don't cause meaningful changes in how a protein functions, and those almost never are things that put people at risk for disease. There are also non-synonymous changes. So this means you end up with a different protein. And so those may or may not have functional importance. And those are often things that become complicated to think about as we analyze genetic test information. And then it's also possible that you can have these variants in parts of your genome that they're not part of the protein, they're probably a part of these regulatory regions. And so these are quite difficult to understand what the functional consequences are without going into the lab and doing specific experiments to test that. And so we all have hundreds and hundreds and hundreds of these. Okay, so in addition to these different classes, different ways that things can go wrong, we think about genetic risk based in some respects on how commonly you see this kind of a change. And so there are really two different classes of mechanisms of genetic risk, things that are rare, and by rare, you can use different thresholds to deem something rare, but generally speaking, these are things that you don't find frequently in the general population. So a threshold that we often use is this particular sequence change is in less than, say, 1 in 1,000 individuals or something like that. These often have large impacts on disease risk. So if you have one of these rare changes in a disease-causing gene, that could mean that you are, 20, 50, 100 times more likely to develop a given disease than someone who doesn't. These usually alter the structure of a protein, although not always, they usually do. In contrast, there are more common genetic variants. So these are things that you find in more than 5% of people in the general population. And so something that's present in 5% of the general population on its own is not going to cause a rare disease. And so that's how we think about things. And so the effect that these common genetic variants have on what an individual person's risk for developing pulmonary fibrosis is a lot smaller. So as opposed to these that make you, 100 times more likely to get disease, these it may be like 10, 20, 50% more likely. So the effect is very different. And these almost never, not never, but these rarely are things that affect the protein, and they're usually found in regions that are, we think of as those regulatory regions, things that don't affect protein coding. Just to try to put that into a picture to think about sort of this, and why I'm dwelling on this is I'll get to in just a second. So rare variants or mutations versus common SNPs. So in gray here, this is, say, the entire population of our country, for example. So in yellow is the basket of everybody who has, for example, one of a common polymorphism that's been associated with pulmonary fibrosis. And then in orange, you have all of the people who have pulmonary fibrosis. So as you can see, a majority, for example, of people with pulmonary fibrosis might have this particular genotype, but most people with that genetic change don't have pulmonary fibrosis. In contrast, rare variants or mutations, which are here in purple, they may only be found in a small number of patients with pulmonary fibrosis, but they're very, very rarely found outside of people who have pulmonary fibrosis. But they make up only a small proportion of all of the individuals who have disease. So again, the reason I emphasize this is that for these common type genetic variants, most people who have them don't have the disease that it's been linked to. So we use different platforms and different approaches to try to target and identify these different types of genes and genetic risk. So for rare things, we can take a given gene and sequence it, so get the whole sequence of all of the bases in it, and we can do that with a number of different platforms. And so the point of this is that for these rare type variants, these are things that can have impacts for patients and for their children. And this is the scenario where, as Janet will talk about somewhat more, genetic testing can be considered and has a role in certain circumstances. In contrast, these common genetic variants, you get through these genotyping arrays and SNP genotyping, but I think because most people who have one of these SNPs don't have pulmonary fibrosis, it doesn't really provide much useful information for any given one person. This sort of thing is hopefully useful in understanding disease biology, but doesn't really have a lot of specific impact on you as a person. And so the reason I bring this up is this is what 23andMe does. This is the kind of information that you get from them. And so why I say that is they'll report out, for example, you have a polymorphism that's been associated with pulmonary fibrosis. Well, it turns out that just based on how frequent all of these common variants are, 99% plus of all people have at least one genotype that's been associated with pulmonary fibrosis. So that sort of information it's informative for understanding the disease, but it does not provide really any meaningful specific information for you as an individual patient. Okay, so thinking about families and what we've learned by studying families, we know that multiple different groups now have shown that about 20% of IPF patients have a relative who also has interstitial lung disease. And as I said, different patterns of disease can be found in these families. And most frequently, these families with disease have a pattern where people in successive generations can get disease. And this pattern is one that's called an autosomal dominant pattern. So that means a single copy of a mutation can be sufficient to cause disease in people. And this is just an example. In black are individuals who had pulmonary fibrosis from a hypothetical family. Rarely you can have pulmonary fibrosis risk mutations pass through mothers' X chromosomes to sons in an X-linked manner. That is quite rare, but has been reported just a few times. And in contrast to cystic fibrosis, for example, where you have to have 2 copies of a risk genotype to give someone disease, it's very rare. There have been just a handful of examples of where sort of 2 copies of a risk gene have been the things that really drove people's risk for pulmonary fibrosis. So that's what I think that the important thing to take away from this is that these diseases are ones where a single copy of a mutation may be sufficient to put someone at risk for disease. But not everybody who has that mutation will develop disease, and I think that that's important. And I think something that we're all very interested in is trying to, on a gene and mutation basis, try to understand how likely is that to cause disease in a given individual. So as I said, 1 copy of a mutation is sufficient to cause disease, but not everybody with a mutation will get disease. So studies now over the course of the last 15 years have as Dr. Garcia talked about earlier this morning, shown that mutations in genes related to telomere biology can be found in somewhere around 25% of families with disease. And the numbers on this vary a little bit from different center to different center. But mutations in this telomerase reverse transcriptase enzyme are the most common. And the rest of these genes are important, but are also only found in a small number of families from what we've seen so far. And so what that has meant is that, genetic risk for pulmonary fibrosis is very different than for cystic fibrosis. And it's very different than for, I don't know if any of you all have friends or family who have pulmonary arterial hypertension. This is another chronic lung disease that can be inherited. 80% of people with pulmonary hypertension with families with pulmonary hypertension have mutations in 1 gene, BMPR2. Pulmonary fibrosis is going to be different than that. And so what that's meant has been a lot of the approaches that we use to try to identify these genes take a long time because there just aren't a whole lot of individuals who have the same change or changes in the same genes. But this list has grown. It's going to continue to grow, and I'm very hopeful that over the course of the next several years, there's going to be 5 or 6 or 7 more that we add to this list, and it's going to keep growing. But there's not going to be one gene that we've missed. So does this affect how people do once they have disease? It appears that it does. And again, applying information from groups of patients to any one patient is something that is difficult to do and is fraught with challenges. But broadly speaking, it does appear that there are differences in outcomes based on some of the genetic risk factors. For example, families who have mutations in those telomere-related genes, people tend to get disease a little bit earlier in those families, maybe 5 or 8 years earlier. So rather than mid-60s is sort of the average age of when someone is diagnosed, it may be more late 50s, mid-50s. And in families with those telomere pathway mutations, it does seem that their lung function may decline a little bit faster, which I guess isn't surprising if people have developed disease a little bit younger. It seems like that disease course is a little bit more accelerated. And so we've tried to look at this and say, is this something that, should we use this information to try to help think about initiating therapy in patients, think about screening unaffected family members? Should we use this sort of information to suggest that someone might benefit from an early referral to a lung transplant center and things like that? This is also a study from Dr. Chad Newton and Christine Garcia. And what they showed in this study, I think, was something that is important, and it is part of the message that I'm really trying to convey. It's that it didn't really matter whether somebody had disease that looked like IPF or hypersensitivity pneumonitis or connective tissue disease, associated lung disease. What really drove their disease course was the underlying genetic risk. So I think that something that we've been talking about, and it was discussed quite a bit this morning, is that we really are in an era where we're going to need to start to move past thinking about these diseases as just based on CT scans and just based on patterns on biopsies, and really think about how incorporating genetic information into diagnoses is something that we'll be able to do. So I'm largely going to skip over this because Janet's going to talk about this somewhat more, but I think naturally a lot of questions come up around how, when, what, if we should consider testing for these gene mutations. And as I say, I'm going to, and I know Janet's going to talk about that quite a bit more, so I'm going to let her address that. Okay, so what have we learned from doing all of this work? Has this led us to any new insights into how these diseases work? And the answer to that is yes. The turnaround from understanding a gene to developing new therapies is not as quick as anyone wants. The analogy that we often use is, the time that it took from finding the CF gene to these new effective therapies in CF was about 25 years. And so we're kind of still on the forefront of this in this disease, and I am hopeful that this is going to move faster. But so the first genes that were discovered in families with pulmonary fibrosis are mutations in genes related to surfactants. So surfactant is a liquid that's made by these cells called your type 2 alveolar epithelial cells, and surfactant has lipids in it, and it has proteins in it, and it's these proteins that essentially keep those lipids fluid enough so that your lung doesn't turn into butter, because the lipids are normally solid at room temperature, at your body temperature. So these are things that help essentially keep our lungs open. And it turns out that these particular proteins require a lot of processing to work right. And mutations in these genes don't actually tend to affect the end product. What they do is they affect how it gets processed, and they impair its ability to get processed. And so what we and a number of other groups have shown is that what happens then is if you can't process these proteins properly, they start to build up in cells, and they activate these stress response pathways that lead to injury and dysfunction and of the cells in the lung epithelium, so the lining cells of the lung. And it turns out that phenomenon is actually not unique just to patients with those rare mutations, but is actually quite common in the lung epithelium of patients with IPF and other forms of interstitial lung disease. And so it's become recognized that there's actually a lot of other ways that you can activate that pathway. For example, cigarette smoke can, low oxygen tension can. So this is something why I think Dr. Kaminsky earlier was mentioning you should wear your CPAP because we know that low oxygen can activate these sorts of pathways, and so can recurrent injury and things like that. And so multiple groups now, including Jeremy Katzen, who was one of the poster award winners last night, at the reception have developed these really exciting new animal models that we can use to try to start to test specific therapies in models with genes and mutations that we know cause disease in people. So I think that this is an incredibly exciting opportunity. What's also really interesting about this pathway is that it turns out that this particular pathway is also involved in how you secrete mucus. So this is sort of a stress response pathway that cells use when they have to secrete a lot of a thing. And so what it appears is that some of the same factors that mediate the cell injury in these models also are things that regulate mucin production, and we're starting to think that it may be sort of the same mechanism that the common MUC5B gene is acting through, just in a different cell type, which I think is very interesting and very exciting and suggests that this pathway is really an exciting therapeutic target. I spent a few minutes talking about genes related to telomere biology, and Dr. Garcia discussed this, I think, at length earlier. But what we've really learned from studying models of these telomere pathway mutations is that they activate these injury DNA damage signals, and that tells cells to stop growing, stop proliferating, and it makes them start to secrete a whole variety of factors that can both try to promote injury repair but also promote fibrosis. And so we think that this is sort of the mechanism as to how these telomere-related mutations act, is that they make cells more likely to have these sorts of responses in the setting of injury. And so I think the implications of this really are considerable. So this particular UPR activation, this unfolded protein reaction, is a common feature in both the alveolar and distal airways in idiopathic pulmonary fibrosis. And there's multiple companies now that are working on developing small molecules and new targets to try to abrogate the pathologic signaling of these genes. And then second, as we heard some earlier, there's also a number of companies that are interested in trying to get rid of or reprogram these senescence type programs that we think are related to how these telomere-related genes act. Okay, last couple minutes, I just wanted to finish up with a little bit of data that we have recently put together that I think helps us inform what we should be doing and thinking about people who have a family history of pulmonary fibrosis. So we've had a study going on now for almost 10 years that we call the early IPF study. So this is a cohort of people who share half of the genes with a familial interstitial pneumonia or familial pulmonary fibrosis patient. So their siblings or their children. And we are enrolling people when they don't have any known disease and don't have any known symptoms. And so what we're doing is then inviting them to come back every 5 years with CAT scans and blood draws and doing a variety of things to try to, one, understand how likely is it for people to develop new changes in their lungs, how rapidly things might progress. Are there ways that we could predict that? Are there ways that we could target this for early intervention? And so what we've learned so far is that perhaps not surprisingly, based on what we know about families and risk for disease, a lot of folks who don't have symptoms have some mild changes. These aren't sufficient to say someone has pulmonary fibrosis, but almost a quarter have mild changes on CAT scan, even without symptoms. And we've been working on trying to identify, okay, what are things that make someone more likely for this to happen? It clearly is more prevalent, it's more common as people get older, and at least in our study, it's very rare in people before age 50, and then the frequency increases progressively from there. It doesn't seem like it makes a difference if you're a man or a woman, but there are a few things that we've started to recognize that are modifiable, that are risk factors for this. For example, smoking, which again, I don't think should be incredibly surprising, but it really does emphasize that if in people who have a strong family history, smoking cessation is 1, 2, and 3 and the best things that they can do for themselves. We've also seen that some of these genetic risk factors may help us predict who is likely to, and other blood biomarkers like telomere length that was mentioned earlier may help predict who is more likely to have these and not. Another thing that has been really interesting that we've observed in this is that people who, so everyone in this study took a survey questionnaire about occupational and environmental exposures before they entered the study. And so while many of the epidemiologic studies that have tried to link different environmental exposures to people's risk for disease have kind of been, they've been confounded a little bit because of how people, they've been done looking back instead of looking forwards. And so we did this in a looking forwards perspective way. And in spite of that, found that people who had reported, in the past exposure to mold, birds in the home, lead exposure, and exposure to aluminum were really significantly increased in people who went on to develop mild changes in their lungs. And so I think that the implication of that is that using that sort of information, knowing what sort of things people may be at particularly high susceptibility for, that allows, that empowers people and their families to make choices about, where they live, what sort of work they do, things like that, that we can't change our genes, but we can change where we are. And so I think there's real opportunity that comes from that. The other thing that we've recognized is I think a real natural question is, well, do these early changes change over time? And the answer that we've learned so far is that it appears that they do. If people have early changes, we've looked at this over the course of about 5 years now in about 100 people. And if you have early changes, most of those do progress. They do increase somewhat over time. But it's pretty slow, at least in this group that we've been looking at. And we've been working with radiologists who have some very exciting quantitative imaging techniques to try to put a number on that. And in these small numbers, it has looked like sort of the change that we've observed is affecting somewhere around 1% of the lung or something like that. And so the reason I think this is exciting is that it means there really is an opportunity for early intervention, even in people who have very mild changes. And I think that presents a lot of promise. So I'm going to close with just a couple of questions that I think about a lot and don't have all of the answers to, but we can talk about this more perhaps in the question session. Is should asymptomatic people with a strong family history undergo some sort of screening? If so, when, how often? And similarly, should people undergo genetic testing, and what about their family members? And I think that is probably the right way to segue into to Janet's talk. So I look forward to discussing this more with you guys in a few minutes.